So, welcome students to the second tutorial session, to the second problem solving session of this NPTEL course, Introduction to Cell Biology. Um, so, in the second week, uh, the focus had been primarily on the next part of the central dogma, which is transcription, translation, and protein folding and the folding of the linear polypeptide chain into protein secondary structures, tertiary structures, quaternary structures, and understanding the forces, the thermodynamics of protein folding, as well as various different techniques that are used for determining the protein structures, the protein dynamics. So just like the previous tutorial session or the problem solving session, this time also I will run you through first through a summary of whatever has been taught more or less in this second week of the course, and then definitely you can ask your questions, which I'll be trying to address, as well as during this summary session. If you have any questions, you can also stop me. Feel free to ask. Please, following the chat is not so easy in the real time, so it will be good if you can ask it by unmuting yourself. Okay, so in the central dogma, we had seen the flow of genetic information, right? So I have not included the central dogma scheme here, but I hope that you remember that DNA is a primary repository of biologically important genetic information. The DNA is capable of replicating itself using the DNA-dependent DNA polymerases, and from the DNA by uh, transcription using the DNA-dependent RNA polymerases, several different RNAs are made, right? For example, mRNAs, tRNAs, rRNAs, and various other types of RNAs. Out of them, mRNA or the messenger RNA is the RNA which is synthesized within the nucleus from DNA by transcription using the RNA-dependent RNA polymerases. Now, this first mRNA transcript that is formed is actually called a pre-mRNA, because typically the genes have two kinds of sequences. One are called the exonic sequences and one are called the intronic sequences. Now, typically the intronic sequences don't code for the amino acids that are present in the proteins. So in this pre-mRNA, which consists of both exons and introns, this pre-mRNA is actually from this pre-mRNA, the intronic regions are cut out, and only the exonic regions are stitched together to form the mature mRNA. And this process is actually called splicing. So the introns are spliced out. The intronic regions which are not coding for any amino acid sequence of the protein are actually spliced out within the nucleus, and the exons are joined together. The blue exons are joined together to form the mature mRNA, which then comes out to the cytosol where the ribosomal machinery is present. The ribosomal machinery then binds the mRNA and reads the nucleotide sequence information present in the mRNA, codon by codon, that is 3 nucleotide at a time, and incorporates one particular specific amino acid residue for a particular codon, right? To form a linear chain, to polymerize amino acids by covalent peptide bond, to form a linear polypeptide chain, right? Which eventually undergoes folding to form your proteins having a certain structure and that perform a certain function. So that completes the central dogma that DNA harbors the biologically important genetic information from the genetic information bank, which is the DNA. mRNA transcript is formed by this RNA polymerase, which is a DNA-dependent RNA polymerase, by a process called transcription. Then the mRNA transcript comes out from is actually spliced within the nucleus to join only the exons and splice out the introns. And then this spliced mature mRNA comes out of the nucleus to the cytosol, where this nucleotide information of the mRNA is translated into amino acid sequence information of the polypeptide chain, which eventually folds into the proteins having a certain three-dimensional structure, which is also important in terms of performing their functions, the respective functions. So now let's look into the nitty-gritties and the step-by-step sequence of events of transcription. That is formation of a mRNA transcript from the DNA by one DNA-dependent RNA polymerase. So just like in DNA replication, in transcription as well, like for example, in DNA replication, we saw that DNA replication starts from the origin of replication. In case of transcription, what happens is that also you can appreciate from this architecture of a gene, which encodes for an mRNA in this case, and eventually the mRNA encodes for a protein, is that there are several upstream elements. Upstream means DNA sequence elements which lie before this genetic element. So these upstream elements, which are called the promoters. So promoter is also a DNA sequence which actually is recognized by several proteins that eventually bring your RNA polymerase, which is the transcription machinery, to the beginning of the gene, so the transcription can take place. So, and these proteins, the proteins which recognize these DNA elements or the promoter elements are called the sigma factors. There are several transcription factors. These are the sigma factor, for example, which recognizes this promoter, which binds this promoter element of the DNA, and it helps the RNA polymerase to dock to this site of the DNA, so that transcription can begin, right? So, in case of transcription, so these sigma factors, several transcription factors, they recognize this promoter sequence of the DNA, and they allow the RNA polymerase to bind to this sequence. Now, RNA polymerase also performs the helicase-like activity. It uses ATP energy, it unwinds the two strands of the DNA double helix, and then it reads only one of the two strands as a template strand and forms the mRNA transcript. Okay, so it binds to the promoter and it unwinds the DNA double helix. So this is a part of the initiation. Then what happens is that what it will do is it will read only one of these two DNA strands. For example, this DNA strand. Okay, in again. So the polymerization can, since we have already learned about the chemistry of polymerization of DNTPs, so we know that the DNTPs can only be polymerized in 5' to 3' direction, because the 3' hydroxyl group will act as the nucleophile that will attack the phosphate of the C5, and the phosphodiester bond can be formed. So this RNA polymerase, if you look that this is 5', this is 3', so it is reading this strand. What it will do is there will be again a small primer, and this will be the 5' end, this will be the 3' end, and then it will elongate in this direction, right? It will continuously open this. So just because of replication, there was a replication bubble. Here it will be a transcription bubble. There will be continuous unwinding of this DNA helix as the RNA polymerase proceeds in this direction, and it keeps on elongating and adding new DNTPs in a complementary base pairing manner, letter by letter. For example, if it is a C, then it will add a G. If it is a, it will add a. But in this case, you have to remember that in RNA, in case of thymine, you have uracil. So if it finds one A, so it will not incorporate thymine, it will incorporate uracil, right? And in case of this transcript that is being formed by the RNA polymerase, this will be an mRNA transcript, which means that this will be formed as a result of addition of not deoxyribonucleoside triphosphates, but ribonucleoside triphosphates, right? That is, the sugar will be ribose. So, yeah, so I hope that this will be useful in appreciating how transcription elongation happens. So after, in the initiation, the RNA polymerase had already bound to the promoter region. It had unwound the DNA. and it had started reading one of the two DNA strands as a template. And what happens is this bubble will be called the transcription bubble. So, and this is the direction of the polymerase movement, right? Here from the left to the right. And so basically what it is doing, it is taking the lower strand as the template strand. It is polymerizing the ribonucleoside triphosphates from the 5' to 3' direction, adding new nucleotides, having ribose as a sugar base. And what is happening? So there is continuous unwinding in this end and continuous rewinding of the DNA again back to the double helix in this end, right? Behind the um, transcription bubble. And this extension continues, but not infinitely. This extension will continue, and this transcript will keep on coming out of this RNA polymerase DNA complex from one exit site. This will continue only till the RNA polymerase reaches one particular sequence of the DNA, which is the transcription termination sequence, right? Where this entire assembly of this pre-mRNA, DNA, and the RNA polymerase will disassemble, and everybody will fall apart. And then this pre-mRNA will be freely present in the nucleus and will be available for the splicing factors, the spliceosomes, to come in and splice out the introns to form the mature mRNA. So the left-hand side, what is presented is the chemistry. So I have already mentioned that in case of RNA, the sugar is not deoxyribose, the sugar is ribose. That is, if I have a 2' hydroxyl group, um, and thymine, instead of thymine, you have uracil in case of RNA. And the polymerization chemistry remains the same. That is a 3' hydroxyl group. You can see here that the 3' hydroxyl group will be acting as a nucleophile, which will be attacking the 5' phosphate, which means the phosphate that is attached to the 5' carbon of the incoming new DNTP, not DNTP. See, this you have to be careful, not DNTP, but NTP, that is ribonucleoside triphosphates. So this 3' OH will attack the 5' phosphate of the incoming new ribonucleoside triphosphate and leading to the formation of the phosphodiester bond, therefore ensuring elongation of this RNA transcript from the 5' to 3' direction, right? RNA transcript will also be formed in the same direction, is 5' to 3'. Okay. So do any of you have any question regarding this transcription, initiation, elongation, and termination? Okay, so let's move ahead. Now we will look very shortly into the process of splicing, um, the phenomena by which these spliceosomes, which are ribonucleoprotein complexes, that is complexes having both RNA enzymes as well as proteins, which are called the spliceosomes, which come together, which within the nucleus work upon the pre-mRNA, right? The mRNA transcript that is formed from the gene or from the DNA, having both the exons and introns. So upon which the spliceosomes work, and they cut off the introns and join together only the exonic sequences to form your mature mRNA, so that it can encode for the for the certain amino acid sequence of the proteins. So how does that happen is that, as I already mentioned, that there are ribonucleoprotein complexes. There are these proteins which are made up of both proteins and RNAs. So they bind at different sites. So they actually bind at this exon intron junctions, right? These components, these splicing components of the spliceosomes, they bind at these exon-intron junctions. And what they do is that because of this protein-protein interaction, then because of this protein-protein interactions or the interaction between these different subunits or components of the ribonucleic of these splicing factors, they come together and causing the looping, right? Causing the looping of the intron so that the exonic ends, right? It causes the looping of the intron so that the exonic ends, these yellow are your exons, the exonic ends are located very close to one another spatially, right? Now what they do is, the first thing that happens is, for example, so this green is your intron. Now the left is the, for example, just for the sake of understanding, consider that the 5' is the left of this. This is the 5' of your intron, and this is the 3' of your intron. Okay. So what happens is that the splicing factors, the spliceosomal complex, they first cut the 5' end. Okay. The 5', they cleave the one phosphoester bond at the 5' end of this intron. And what they cause instead is a 2' 5' phosphodiester linkage here within the intron, causing a closing, a covalent closing of the intron, right? And forming of this intron lariat. See, there is a circular covalently closed. This is a circular covalently closed part of the intron. But this bond is actually the 2' 5' phosphodiester bond that we also discussed in the previous class. This is very interesting. And then there is a tail. Okay, but still this is not yet cleaved from this exon. Then the next part, in the next step, what happens is that this 3' end of the intron gets cleaved from the exon, and this intron lariat, this is called the intron lariat, which is because this is the circular covalently closed part of the intron, but also having a 3' tail. So then this lariat is released, and eventually these exons are joined together to form a continuous mature mRNA, which is consisting of just the exonic regions and not the intronic regions, right? So that it can encode for the desired amino acid sequence of the polypeptide chain, so that it can fold into a certain into the respective protein structure, which can perform a certain function. It can be catalytic activities, it can be different activities. But however, I mean, this is the primary concept. But one thing I will like to mention in this case, that it is not always the case that introns don't encode for amino acid sequences of the proteins. There is something which is called alternative splicing. So in some cases, there is a differential splicing of the introns. So what does that mean is, for example, I will mention in this slide that, for example, in some cases it happens that in one mRNA transcript, there can be different mRNA transcripts, different mature mRNA transcripts that are formed from the same pre-mRNA. And that happens because, for example, one mRNA transcript is formed where all the introns are cleaved, where all the introns are cut out and only the exons are made, right? Which is this one. But also there can be combinations. For example, there can be one mRNA transcript, mature mRNA transcript, where only this intron is cut out and the rest is joined together. Or where maybe these two are cut out and all the rest are joined together. Or maybe these two, the first and the third, are cut out introns and the rest are joined. So you can, if you are interested, you can also read more about that. This actually leads to formation of different protein isoforms as well, because these different mRNAs have different nucleotide sequence, as you can imagine, and therefore they lead to different amino acid sequences of the proteins having considerable homology or similarity between one another, but also quite stark differences among themselves. So leading to different protein structures, different proteins, different protein isoforms coming from the same gene, right? So this thing called alternative splicing, a phenomena called alternative splicing, allows you to get different protein isoforms from the same gene that can encode for different mature mRNAs. Okay. So now moving forward, so basically, since we have already gone through transcription and splicing, so we have the mature mRNA, transcript, which has a nucleotide sequence and can encode for the amino acid sequence of the proteins, right? So how will that happen? This mature mRNA comes out of the nucleus into the cytosol, where the translation machinery, that is the ribosomes, are present. So what do the ribosomes do? And now
Sir,
this, in this
sir,
respect, which, ah,
hello? Sir, the UTF that is displayed on the previous slide, what is that? U2AF on the previous slide.
uh, sorry, which one? U2A. Oh, okay. This is just a ribonucleoprotein complex, which is a part of the this spliceosome. There are several components. It's a multi-component complex. So this is the name of just of a protein, which is one of the components of the spliceosome.
Okay, sir. Sir, I have a question for the uh, lagging
Hmm.
strand. Can I tell?
Yeah, yeah. You can ask.
Yes, sir. The lagging strand forming the Okazaki fragments. Why it is
Hmm.
forming discontinuous?
Yeah. So the thing is, I will just try to draw and let you understand. So the thing is that, for example, this is your replication fork. Okay. This is your replication fork. So So kind of this is your replication fork, and your helicase is moving in this direction. Okay, from right to left, let's say. Now, um, so right now, so in this case, which strand will be your leading strand?
So the down strand means
No, the leading strand. The leading the
yes.
leading strand will be the upper strand, because this is your direction of the movement of the helicase or the replication fork. So, and you see that the so DNA polymerization will always happen in the 5' to 3' direction, right? So in the upper strand, this is the 5' to 3' direction, which is in the same direction as the movement of the helicase. So this will be your leading strand, the upper strand. Whereas the lower strand, if you see, there the direction of polymerization, which is again, which will always be the constant, 5' to 3', right? But this is in the opposite direction. This is in the opposite direction to the direction of movement of the helicase. This is the direction of movement of helicase, which is from right to left. But for the lower strand, the DNA polymerase has to move from, in this schematic, if you see, then for the lower strand, the DNA polymerase has to move from the left to the right, right? You agree? So for the lower strand, which will be the lagging strand, what I'm trying to say is that in this schematic at least, the DNA polymerase has to move in the opposite direction. Um, Opposite to the direction of movement of the replication fork and the helicase DNA polymerase complex machinery. So in biology, what happens is this is not possible, right? So to make it possible, what happens is that for this lower strand, you cause a looping of the DNA. This lower strand gets looped, right? This lower strand gets looped, and then what happens is that the DNA polymerization has to happen in the same direction in which the in which the helicase is moving. So here one primer is formed, and then the DNA polymerase elongates this to form one fragment, right? Now, by the time it has, so there are two things. One thing is that the processivity is lower. Because of the DNA looping, there is a considerable amount of torsional stress that is created, and the DNA processivity is lower. That means that DNA can polymerize, for example, 50 to 60 bases or 100 bases, much lesser number of bases at one go on the lagging strand as compared to the leading strand. So it forms a short fragment, but after that the DNA gets separated. This looping is not a very comfortable thing. Somebody is kind of holding it together, but After the more is the length, the more stress it generates, and the more difficult it becomes to hold it together. So the DNA actually flips out. DNA comes out of this DNA polymerase complex. So after the formation of the short fragment of the fragment of the new lagging strand, the DNA it actually comes out of this DNA polymerase machinery. So then what happens is, and by the time you can realize that the helicase is continuously going ahead in this direction, right? So new part, I mean, there is now more part of this strand which has been unwound by the helicase, which also needs to be replicated. So then what happens is the primase enzyme comes and forms a forms a new RNA kind of not RNA, new oligonucleotide primer in this part, and the DNA polymerase again binds to this region and starts by there is again a DNA looping, and the DNA polymerase starts to elongate that, right? But again, after it has elongated, for example, 50-60 bases, the loop becomes very long enough, and that much long enough that it again snaps out of this DNA polymerase DNA complex. So again, the DNA polymerase is cut off from that fragment, so it cannot extend or polymerize it anymore. So that's why on the lagging strand, this DNA polymerization or the elongation is actually discontinuous, leading to the formation of short DNA fragments, which are called the Okazaki fragments. This problem is not there for the leading strand because there's no DNA looping is needed. So the problem arises because DNA looping is the DNA needs to be looped. This lagging strand needs to be looped so that DNA polymerization can happen from 5' to 3', but also in the same direction in which the replication fork is moving or the helicase is moving. But with this DNA looping, what happens is the more is the length of this loop, right? The more is the stress or more difficult it becomes for the DNA polymerase helicase machinery to hold it together. So it snips out, it snaps out, it comes out of this from the grip of the DNA polymerase. If it comes out of the grip of the DNA polymerase, then it is not able to synthesize more than a certain length, more than a certain number of nucleotides. So that's why it becomes discontinuous. So that's why it needs quite a bit of, start stop. Like there is a start, there is synthesis of 60, there is polymerization of 60, 70, 80 bases, and then it again snips out. So another round of initiation is needed. Another round of this primase mediated oligonucleotide formation is needed. The DNA polymerase again binds, again polymerizes, but it again snips out. It's a multiple round of start stop kind of thing.
Okay, sir,
Right? Can you
understood.
understand?
Sir, yes, sir, understood. Sir, one question. Uh,
Yeah, sure. Go ahead.
yes, sir, but sir, why we are telling that DNA dependent polymerase or RNA dependent polymerase? Why dependent RNA dependent or DNA dependent? Means it is dependent of DNA or RNA dependent. That's why we are calling DNA dependent, RNA dependent.
Good question. The answer is quite simple actually. So dependence means that, see, for replication as well as transcription, what essentially is happening is that you are polymerizing new DNTPs, right? You are polymerizing, for example, in case of replication, you are polymerizing DNTPs. In case of transcription, you are polymerizing NTPs. What I mean to say is ribonucleoside triphosphates, since it's an RNA. But while doing so, what you are essentially doing is you are copying, right? You are copying some template. So you need some template. The template can be DNA, the template can be RNA, right? So, for example, during DNA replication, so essentially you are copying one DNA to form a new DNA. So that's why it's called DNA dependent DNA polymerase. So that means it follows one DNA strand as a template, and then it polymerizes DNTPs to form a new DNA strand. Whereas, as I gave the example that, for example, in case of RNA viruses, after they infect the cells, the host cells, so within the host cells, there is a replication of these RNAs. So from there, what you can imagine that from one RNA, many more RNA molecules are made, right? So then there it becomes RNA dependent RNA polymerase. So what it means is that it takes one RNA as a template, one RNA molecule as a template, and it synthesizes NTPs to form new RNAs. Now, in case of transcription, what we are saying is that it's a DNA dependent RNA polymerase. What it means is that it takes DNA as a template and it polymerizes NTPs to form new RNA molecules. So that's how this nomenclature works. So here, DNA dependent DNA polymerase, I can understand your confusion. So DNA here, for example, in this DNA dependent RNA polymerase, DNA is not a cofactor that is necessary for the RNA polymerase activity. DNA is just kind of the nucleotide, the nucleic acid nature, the nucleic acid molecule, which it is taking as a template for polymerizing new NTPs to give rise to a different kind of a nucleic acid molecule. Right?
Okay, okay, sir, understood. Uh,
Okay.
the same as that follows for RNA also. RNA is taking as a template, so it is
Yeah.
getting RNA dependent.
Yes. Here they are not cofactors. They are just the dependent. Dependent means which molecule it is taking as a template for the synthesis of which different new molecule.
Okay, sir, understood.
Yeah. Okay. So to proceed with, so yeah. So now we have this mature mRNA which has come into the cytosol, which will be read by the ribosomes, which are the translational machineries, to form the polypeptides, to polymerize the amino acid residues by covalent peptide bonds to form the polypeptides that will eventually fold into the respective protein structures that will eventually give rise to protein functions. But it was a mystery for quite long time that, for example, this mRNA is actually a mRNA has a certain nucleotide sequence. But this was a big question in molecular biology, that how is this mRNA information read? What is the, for example, in case of, for example, you have one coded message from ISI or from raw, and you have to make some sense out of it. So it will be written in some pattern. Only if you know the pattern in which that coded message is being written, you will be able to decode it. So this was a big question in molecular biology, that what is the pattern of writing this nucleotide sequence information in these transcripts, in these mRNA transcripts, that these decoding machines, that is the ribosomes, know. And so how do they interpret? What is the nature of interpretation of this mRNA information to polymerize a certain sequence of amino acids in the polypeptides? And it was Nirenberg, Khorana, and Holly. So it was primarily Nirenberg and Khorana who elucidated this genetic code, the code of storing the nucleotide sequence information in mRNAs that is being read by the ribosomes to form the proteins. And Holly was also a part of that Nobel Prize, who had worked quite a bit on the tRNA, the transfer RNA structure and its molecular details. So how it works, which is quite well known today, is that the ribosome reads three nucleotide bases together. And for one particular set of these three nucleotide bases, it encodes, it actually incorporates one particular amino acid residue. So how does that work? So this set of three nucleotide sequences that we call a codon, and how it actually works is that this transfer RNA or the tRNA, it has one exactly complementary nucleotide sequence, which is called the anticodon. And because of this specific binding of this tRNA having a certain complementary anticodon to uh, the codon of the mRNA, while it is present within the ribosome, this tRNA is able to deliver one particular amino acid to the ribosome. And thus, for each of these codons, the ribosome is able to incorporate a specific amino acid residue in a certain sequence that leads to the specific amino acid residue compositions of the different polypeptides and proteins, right? So, and this tRNA, which we will also study a bit more in the upcoming slides, this tRNA is called amino acyl tRNA, right? What does amino acyl tRNA means is that the amino acid is actually loaded. There are some enzymes which are called the amino acyl synthetases. There are specific enzymes which recognize a certain tRNA having a certain anticodon sequence, and they load a specific amino acid molecule on those tRNAs to form this amino acyl tRNAs. And that is also energy kind of investing process. It's a energy demanding process. You need one ATP energy to form this amino acyl bond, to load this amino acid on a tRNA, to make the charged tRNA. Okay, so let's look a bit more into so there are four different types of nucleotides, A, G, U, and C. And each codon is consisting of three nucleotides. So there can be 64 possible combinations, right? There can be 64 possible combinations, but there are only 20 standard amino acids that are being encoded, that are found in the proteins. So it was found that there is a considerable amount of degeneracy. What that means is degeneracy or redundancy. So what that means is that one amino acid. So you can see that if there, for example, there are 64 codons which are encoding for 20 amino acids, right? So for example, there were 64 codons and there were 64 amino acids. Then most likely what it turns that several different codons actually encode for one particular amino acid, which you can also see. For example, in case of arginine, if you see, or arginine, you can see, right? In case of threonine, you can see, or various other amino acids, you can see that more than one codon. Here there are 6 codons which can encode for arginine. Here there are 4 codons that are encode for threonine. So there is a degeneracy or redundancy. However, in this regard, it is also notable. It is mention worthy to state that how this degeneracy actually happens is that, for example, if you just look at the codons, then this is for arginine, you can see that it will be CGU, CGC, right? If I write them, CGU, CGC, CGA, CGG. So among all of these codons, we can now see now. So basically, we were saying that in the as we were discussing in the earlier slide that in mRNA there is a certain codon. So there will be a tRNA which has the exact complementary anticodon, and it will be binding to that particular codon in a complementary base pairing fashion, the Watson-Crick base pairing fashion, and it will be delivering one particular amino acid residue in response to a certain codon of the mRNA. Then how is it possible that four different codons are, that is, four different sets of three nucleotide sequences are encoding for the same amino acid residue? Are kind of encoding for the same amino acid residue means that are able to base pair with maybe the same tRNA residues, the tRNA anticodons. So it turns out that in for some codons, in some codonic sequences, this third base, right? This third base change is actually tolerable, and it's not a specificity determinant. That is, even if there is a change in this third nitrogenous base of the codon, it is still able to base pair with the specific tRNA, which brings in a particular amino acid residue. And that's why this third base is actually called the Wobble base, right? It's called the Wobble base. Okay. And that allows the degeneracy or the redundancy of the genetic code, where more than one codon is able to encode for one particular amino acid residue. So this third base is not the determinant of specificity. It turns out that the first two bases are the main determinant of the specific binding to a particular tRNA having a certain anticodon. And this mismatch of the third base is tolerated. And even if there is a mismatch of the third base, this perfect base pairing of the first two bases in some codons is capable of delivering the is capable of delivering or incorporating a particular amino acid residue because of the capability of base pairing with the tRNA having the particular anticodon sequence. Apart from that, two other facts are mentioned worthy, that AUG is your start codon, right? The start codon is AUG, and in response to the start codon, always this methionine amino acid residue is incorporated. And there are three codons which do not encode for any amino acid residue. Instead, they encode for they are like the stop signals for translation. So if the ribosome actually encodes any one of these three codons, which are the stop codons, then there is actually no transfer RNA. There is no tRNA which can complementarily bind to these particular codons. So no tRNA having the amino acid residue, no amino acid tRNA comes in. Instead, what happens is the RNA, this ribosome complex gets stalled when it encounters one stop codon. And then the release factors, which are also proteins, they come in and they actually rescue, they dismantle, they disassemble this ribosome mRNA complex. Everything falls apart and translation comes to a stop. So there, these ribosomes get disassembled and you get the mRNA and the polypeptide chain separately in the cytosol. So these are your three stop codons, UAA, UAG, and UGA. Okay, there are three stop codons, UAA, UAG, and UGA. Okay. So, right. Now, let's have a look into the transfer RNAs and the amino acid tRNA synthetases. So, as I mentioned, that this tRNAs, for example, when we were we were discussing about this mRNA, we were always representing this mRNA or which is basically a ribonucleic acid as a linear chain, right? But you always can think of this that the single-stranded RNA can also fold upon itself. For example, why is the DNA a double helix? The DNA is a double helix only because of this complement. There are two DNA strands which are held together by the complementary base pairing because of this non-covalent hydrogen bonds between the purines and pyrimidines of two different strands, right? So after the formation of the single-stranded RNA, there can well be intra-chain interactions, right? Intra-chain hydrogen bonding interactions between the complementary bases within this RNA, which leads to formation of several secondary and tertiary structures, which is a classic case in case of this tRNAs or transfer RNAs. So basically, you can see in this left picture that after the tRNA is synthesized as a result of transcription of the tRNA encoding genes within the nucleus, they form the secondary structures because of the secondary kind of hairpin structures, right? These are the hairpin structures because of the complementary base pairing within the RNA chain, like intra-chain hydrogen bonding, and leading to the formation of these different structures, the anticodon loop, where you have this three nucleotide sequence, which is the anticodon, which is a specificity determinant that can specifically bind to a certain codon of the mRNA and will be encoding will be incorporating one particular amino acid residue in response. And then there are these other loops, T-Psi, CG loop, D loop, having the predominance of dihydrouridine, inosine, right? And most importantly, this acceptor stem. So in this acceptor stem, you can see that there is a 3' CCA, right? there's a 3' CCA, and the amino acid residue which is to be loaded on the tRNA is always loaded on the 3' on this 3' CCA motif, on this 3' CCA amino acid nucleotide sequence. So this is called, this secondary structure is called the clover leaf model. The clover leaf secondary structure of tRNA. And after assuming the secondary structures having these characteristic hairpin loops, then the tRNA further folds into three-dimensional L-shaped structures. Okay, so this is the L-shaped tertiary structure of the tRNAs, where you can also recognize these secondary elements, the D loop, anticodon loop, T size CG loop, and finally the acceptor strand, which is a 3' CCA, where the new amino acid residue, where the amino acid residue will be loaded. Now, also, so for ensuring, so while this translation, you have to always, I think, appreciate this fact that there is a fantastic specificity in how a certain codon is being read by the ribosome, and a certain amino acid sequence, a certain amino acid is incorporated in response. So, but this specificity is actually ensured at several levels. And one of those levels is that you need to have a certain amino acid sequence being loaded on a tRNA having a certain anticodon nucleotide sequence. That is ensured by this set of enzymes which are called the amino acid tRNA synthetases. So these amino acid tRNA synthetases recognize a certain tRNA, okay, a certain tRNA having a certain anticodon sequence, and recognize the particular amino acid residue, the free amino acid, which is to be loaded on this tRNA, right? And then they use one ATP energy. So you can appreciate this fact that loading one amino acid on a tRNA is energy dependent, and that's why eventually translation becomes a very energy demanding process, because formation of each covalent bond essentially is utilizing one ATP energy in the end. Um, so here, this amino acid tRNA synthetase uses this ATP energy to load the amino acid on this tRNA, and by formation of this high energy ester bond. Eventually, if you dig deeper and look into the details of how this translation happens, you will find that the energy that is being released as a result of breaking this high energy ester bond is used for the formation of a peptide bond that links two different amino acid residues of the polypeptide chain by some enzyme which is called the peptidyl transferase, which is present in the ribosomal subunit. Okay, so finally, because of this amino acid tRNA synthetases, what you essentially get is called an amino acid tRNA, right? Amino acid tRNA, which is this one. So here the amino acid residue is phenylalanine. Get the amino acid tRNA, which means a charged tRNA, which has one amino acid residue loaded on the 3' CCA of the acceptor stem, which is loaded via the formation of this high energy ester bond. And this energy will be utilized actually during the translation process for the formation of the new covalent peptide bond that will be linking the two amino acid residues. Okay. So let's now look into the step-by-step, the players involved in the process of translation, as well as the step-by-step sequence of events in translation of the mRNA nucleotide sequence into the amino acid sequence of the polypeptide chain. So the primary machinery of translation is your ribosomes, right? So these ribosomes are ribonucleoprotein complexes. So what that means is these ribosomes are made up of RNAs as well as proteins. And very interestingly, RNA actually constitutes almost 65% of the ribosomes. And many of these enzymes of the ribosomes are actually RNA enzymes. They're called the ribozymes. Okay. Now RNA actually consists of two subunits. These ribosomes consist of two subunits. One is the large subunit and one is the small subunit. Okay. Actually, even these large subunits and small subunits consist of further smaller subunits which assemble together to form these small subunit and large subunit, which eventually come together during the process of translation. So, and For example, if you try to isolate these subunits from the cells, be it bacterial cells or eukaryotic cells, and you do a centrifugation experiment, a density gradient centrifugation experiment, then you, because of the different size and the different effective molecular weight, they get fragmented into they get kind of co-partitioned into different sections, and because of the different effective molecular weights. And because of that, they are, the file is large for this prokaryotic ribosome. It consists, these two subunits are 50S, where S stands for this Swedberg unit, an unit of this mass, which essentially give rise to different centrifugation profiles. Different profiles are different bands, different co-partitioning in the density gradient centrifugation, where you are basically applying centrifugal force. So the heavier it is, the lower it goes. The lighter it is, it stays in with upper fractions. So this large subunit is 50S in case of the prokaryotic ribosome and 60S in case of the eukaryotic ribosome. The smaller subunit is 30S in case of the prokaryotic ribosome and 40S in case of the eukaryotic ribosome. However, together, as you can very well, I think you can imagine that this is not a very additive thing, because the sedimentation depends on two opposing things. One is their effective molecular weight. So more is the effective molecular weight, it will try to sediment lower. But when two things come together, their effective hydrodynamic volume, which gives them buoyancy, that also comes into picture. So these Swedberg units are not additive. This prokaryotic ribosome, the entire prokaryotic ribosome consisting of the 50S subunit and 30S subunit, sediments as a whole as 70S, whereas the eukaryotic ribosome, which consists of the 60S and 40S subunits, sediment as a whole as the 80S ribosome. And what this 5S, 23S, 16S means actually are the further smaller subunits, right? Here it is written RNA, but actually it means RNPs, which means ribonucleoprotein complexes. That is, RNA is associated with proteins, right? Now, this lower image actually shows you a ribosome mRNA nascent polypeptide chain tRNA complex, the actual translation
Oh,
complex. Huh.
sir, can you tell that what is the DA means?
What is DA? Where is DA?
2.7 into 10 to the power 6
Oh,
DA.
DA actually means Dalton. So it's actually an unit of molecular weight.
Okay,
For
sir.
example, in case of proteins, you will come across kilodaltons for whites. Yeah, so it's basically unit of molecular weight. Okay. Yeah. So in this lower panel, what you can see is actually the real-time translation complex, where you can see that the ribosome. So how it happens actually is that once this mature mRNA is present, then the smaller ribosomal subunits first recognizes one sequence element. Okay, recognizes one particular sequence element of the mRNA, which is called the ribosome binding site, and it binds this mature mRNA that has come from the nucleus to the cytosol. After the small subunit binds to this mRNA, then the large ribosomal subunit binds to this complex of small ribosomal subunit and the mRNA. Now it binds in such a manner that these ribosomes, after this formation of this complete complex, that is the mRNA sandwiched between the mRNA sandwiched between the small ribosomal subunit and the large ribosomal subunit. Once that is done, then it happens in such a manner that the start codon, that is the AUG, is present here. Okay, this site is called the P site or the peptidyl site. Okay, so this is the site where the enzyme peptidyl transferase is present, and what it does is it takes the amino acid from the tRNA, which will be present here, and transfer it to the amino acid that is present on the next tRNA, right? That is by the formation of a covalent peptide bond. So this is the P site or the peptidyl site. This is your A site or the amino acyl tRNA site. Okay. And this site is actually your exit site from which a nascent elongating polypeptide chain, right? As the ribosome moves and polymerizes more and more amino acid residues via the covalent peptide bond, the E site is the site of the large ribosomal subunit through which this nascent polypeptide chain, the elongating emerging polypeptide chain comes out. So when the first this translation complex is formed, that is, there is the mRNA, small ribosomal subunit, and the large ribosomal subunit. The start codon will always be present in the peptidyl site, and one more amino acyl tRNA residue will be present in the amino acyl tRNA site. Okay, and okay, so we will also see this better in the upcoming slides. So in this in this lowermost diagram, what you can see is that, for example, this is the P site, right, where already this tRNA is present, which uh, which when it came, it had one particular amino acid of its own because it was an amino acid tRNA. Then this peptidyl transferase enzyme, what it did is it took away this entire chain from this tRNA and put it onto this tRNA, right? And then what it did is this ribosome moved one codon ahead. So this will again come to the P site, right? And this site will be empty. A new tRNA will come in this site, and this by the time this tRNA will be empty, it will be present in the E site, and it will get out of the translation complex to get recharged once more, to get recharged by these amino acyl tRNA synthetases, which are present with the cytosol, which will bind this empty tRNA, which will bind the respective amino acid residue, and will load the amino acid residue on the tRNA to again generate a pool of amino acyl tRNA. So this cycle keeps on going. So the supply chain is never off. So we will gradually try to understand this, the steps of translation even in more details. So as I already mentioned that when this mRNA mRNA comes to the cytosol, first the small ribosomal subunit, right? The small ribosomal subunit will bind, then the large ribosomal subunit will bind, and this will give you the entire ribosomal complex. Now, within that, you will find initial that at the P site, your start codon, or the peptidyl site, your start codon will be present, right? Which is AUG. And at the A site, the next codon will be placed. So, for the start codon, always this one particular tRNA, like methionine tRNA. A tRNA having methionine and this codon which binds to AUG, which is having this anticodon basically, which is complementary to the start codon AUG, will come in. Now the peptidyl transferase, what it will do is, the methionine is bound here with this high energy ester bond, and here for example, the next amino acid residue is also bound. Peptidyl transferase will transfer methionine from this amino acid tRNA to this particular amino acid, which is bound to the next tRNA by the formation of a covalent peptide bond between these two amino acid residues, that is methionine and tyrosine in this case. It can be methionine and any other amino acid residue, which depends on the protein sequence or the polypeptide sequence. Now, after this is done, what will happen is that, so you can I think visualize that this tRNA is now empty. The methionine tRNA that had initially brought methionine is now empty. It does not have any amino acid residue on its neck. The entire methionine tyrosine is now attached to this tRNA, the second tRNA, the tRNA, right, which is bound via the complementary anticodon-codon binding. Now, what will happen is, which you can see in this case, is that this ribosome will move in this direction. Now, this direction is also important. Ribosome will read the mRNA always from 5' to 3' direction. Okay. And this polypeptide chain, okay, the polypeptide chain that is getting synthesized will always be getting synthesized from the N to C, right? From the N to C direction. That is, for this methionine residue, the N terminal will be free. There will be free NH2, okay? Whereas here the tyrosine's COOH, the C double bond O, single bond O, if you can imagine, is currently is in the form of an ester with the tRNA, but eventually when the polypeptide will be released, this will be free. So this ribosome will always keep on reading the mRNA in 5' to 3' direction, and the polypeptide chain, the nascent new polypeptide chain, will always be synthesized in N to C manner. Okay. So now you can see that the ribosome has moved one step, right? One step means one codon. So what has happened is this empty tRNA, okay, which doesn't have methionine anymore, is now present on the E site, right? E site of the ribosome. That is the exit, which is basically it comes out of the ribosome. It is released into the cytosol so that the amino acid tRNA synthetases are again able to bind this empty tRNA and again load a methionine on the neck, on the 3' CCA of the acceptor stem. Whereas this A site now gets empty, now gets vacant and is available for one new tRNA having the anticodon region which is complementary to the codon, right? And thus bearing and bringing in one particular amino acid residue that this codon encodes for in this place, which you can see in this next figure. The next tRNA having the anticodon region, having the anticodon region which is complementary to the codon, and thus bringing in one particular specific amino acid residue which is attached to 3' CCA of the acceptor stem of the tRNA into this translation complex. And again, this will this will keep on happening. What will keep on happening is that this methionine tyrosine will be now transferred by the peptidyl transferase to the next amino acid residue that is coming in by the formation of a peptide bond. Again, the ribosome will move one step forward, that is in this direction, that is from 5' to 3', and the nascent polypeptide chain will keep on growing. New amino acid residues will be kept on adding in the N to C manner, right? So, but this will again, so as you can see that there has been an elongation of the polypeptide chain, and the nascent polypeptide chain has grown. But this will not go on infinitely. There will be one state where this ribosome will be, while this ribosome will encounter one stop codon, right? One stop codon at its A site. Now there will be no tRNA that can bring one particular amino acid to this site. Then what will happen is the release factor, the ribosome release factors instead will come in and release these, disassemble, they will dismantle these stalled ribosomal assemblies. So the large subunit will separate, small subunit will separate, the mRNA will separate, the polypeptide chain will separate, as well as the tRNAs will separate. Now we will again come back to the square one, kind of to that stage where you began with, that you have this mRNA, you have these ribosomal subunits, you have the empty tRNAs, which can be again reloaded with the amino acid residues. But now you have this nascent polypeptide chain, right, in your cytosol, which will now fold into its respective structure so that it can perform its respective function. And as we will see, that is what we will be discussing in the next slides, that how this nascent polypeptide chain, which is basically just a linear polymerized sequence of amino acid residues, right? It's just several amino acid residues being polymerized by the covalent peptide bond. Still doesn't have any structure, but gradually it will start adopting some secondary structures, and eventually the secondary structural elements will also arrange in a certain three-dimensional manner to form these tertiary structures. And formation, after formation of these tertiary structures, even several different subunits can come together and get organized and assembled in a self-assembled in a certain manner to form even the quaternary structures. And in a process which can also be helped by other factors like cofactors, right? There can be, for example, you must have also seen the heme cofactor and hemoglobin, right? There can be various other cofactors, vitamins acting as cofactors so many times. Yeah, there can be metals that are acting as cofactors. Right. So, so this is how translation um, uh, via this ribosomal machinery finally gives you this linear polypeptide chain from the mRNA nucleotide sequence. Linear polypeptide chain having the amino acid residues polymerized by covalent peptide bonds formed by reading the nucleotide information that was present in an mRNA transcript. Right. So next up, we have protein folding. That after transformation, after translation, you have the linear polypeptide chain where you have these amino acid residues that are joined together by the polypeptide, by this peptide covalent bond. But this amino acid sequence, right, is for every different polypeptide chain will have a very different amino acid sequence. And actually, the amino acid sequence itself has the amino acid sequence information itself is the information that governs what structure that polypeptide will eventually assume. What structure the polypeptide chain will be stable in, because there are different secondary protein structures that this uh, linear polypeptide chain will assume. So linear polypeptide chain primarily has just the covalent as, linkages. For example, the covalent peptide bond and another covalent bond it can have just after translation is the intra-chain cysteine disulfide bonds, right? The disulfide bonds that are found between the two sulfadyl residues of the cysteines. But gradually, the non-covalent interactive forces like hydrogen bonding, hydrophobic interaction, electrostatic interactions, that is negatively charged residues interacting with positively charged residues, those salt bridges, dipole-dipole interaction, cation-pi interactions, those step in that lead to further structuring of this genetic polypeptide chain into secondary structural elements like alpha helices and beta sheets, as well as other things like beta turns, loops, etc. Eventually, what happens is the secondary structural elements also interact among themselves, right? Leading to leading to a certain three-dimensional orientation or organization of these secondary structural elements of the proteins, leading to the formation of the well-defined three-dimensional structures, which are called the tertiary protein structures. And eventually, what can happen is that so each of these like two, three, four different subunits, protein subunits, each of them having their own three-dimensional tertiary protein structures, can come together, right? Can come together to form even higher assemblies, which are called the quaternary protein structures, which where two, three, or more. The number depends from protein to protein, where different protein subunits can come together to form a higher assembly. An example being hemoglobin itself, which consists of 4 subunits. Right. So the just to touch upon a bit of details. So I mentioned that the amino acid sequence of the polypeptide chain itself has the information that is necessary to dictate or govern that how the folding will be, right? Because effectively it's the amino acid interactions, the intra-chain amino acid interactions that drive the folding of the linear polypeptide chain into a folded polypeptide chain and gives the folded polypeptide chain or a certain protein structure the stability that keeps it in that particular structure, right? So to understand that, first you need to appreciate that there are different kinds of amino acid residues. So some are positively charged amino acid residues like lysine, arginine, also histidine at a certain pH. There are negatively charged amino acid residues like aspartic acid, glutamic acid. There are aromatic amino acid residues, right, which can form these pi-pi interactions and cation-pi interactions. So like tryptophan, tyrosine, phenylalanine. There are these non-polar hydrophobic amino acid residues like isoleucine, leucine. There are very unique amino acid residues like proline, which are present in the beta turns, which are kind of helix breakers, and some amino acid residues like glycine, which have very less constraint and can fit in many, provide flexibility to the polypeptide chain. So if you just look at the different types of amino acid residues, then you can appreciate these different types of forces like hydrogen bonding, the hydrophobic interaction, where the hydrophobic residues try to hide from the aqueous environment or the water molecules to ensure higher entropy or freeness of the water surrounding water molecules, and enthalpic interactions between the hydrophobic residues, which try to hide within the structure, protein structure, the folded protein structure that stabilizes a certain protein structure. So there are hydrophobic interactions. There are this hydrogen bonding interactions, intra-chain hydrogen bonding interactions. There are electrostatic interactions, the negatively charged protein interacting with the positively charged protein. Salt bridges, where ions also help in forming intra-chain interactions. And obviously, there are these dipole-dipole interactions, there are cation-pi interactions, pi-pi interactions, van der Waals interactions stabilize this linear polypeptide chain into a certain structure. And definitely, so the structure is governed by the amino acid sequence of the polypeptide chain. And also, often this 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 disulfide bonds, right? The disulfide bonds which are formed between the sulfhydryl groups up to cysteine residues. So this can be intra-chain, this can be inter-chain. For example, in many cases, inter-chain disulfide bonds can also be formed, especially while formation of these quaternary structures or higher self-assemblies. And proper intra-chain disulfide bonds are a major stabilizing force in protein structure. There are several different chaperones or other S proteins which assist the formation of proper disulfide bonds, intra-chain disulfide bonds. Without which the particular protein folding is not will not be possible. Okay, so these are kind of your two primary major protein secondary structural elements. There is the alpha helix and the beta sheet. So the alpha helix, so actually there are very characteristic amino acid sequence compositions that predispose the formation of one particular secondary structure over the other. Those amino acid compositions which enable this a particular pattern of hydrogen bonding, the N to N plus 4 hydrogen bonding. That is a hydrogen bonding happening between one particular amino acid and the amino acid which is 4 amino acid residues later, right? Than that. So N to N plus 4 hydrogen bonding is the formation of alpha helices. Okay, and for alpha helices that have a certain characteristic, this distance between amino acid residues and pitch, just like the DNA double helix, that there is, for example, 0.34 nanometer, 0.34 nanometer distance between two bases. So there's a kind of that that kind of characteristic pitch that between two amino acid residues is that much distance. So those amino acid sequences that allow N to N plus 4 hydrogen bonding typically have higher propensity for formation of alpha helices. However, there are also different kinds of helices. For example, collagen helix is quite different than that of alpha helix, having its own characteristic hydrogen bonding. So in some helices, which is N to N plus 3 hydrogen bonding, but the classic alpha helices are N to N plus 4. In beta sheets, what you have is the hydrogen bonding between two kind of parallel or anti-parallel sheets of amino acid residues, that are horizontal uh, kind of hydrogen bonding. And between the two sheets lie these beta turns or loops. Okay, connect them. Now we will just discuss a bit about some this concept of the dihedral angles, and because this is one important thing that or constraint that along with the amino acid which actually governs the protein folding. So basically, so first we have to look at the peptide backbone, the covalent peptide bond that joins together amino acid residues. So if you look that this is your N terminal, right, having the NH3. This is your C terminal having the CO minus. Right? This is your C terminal. Now, after that, this carbon is your C alpha. This is your C alpha carbon. This C double bond O carbon is your C prime carbon. This is your N, and this is the peptide bond, right? This is the peptide bond. In a polypeptide chain, we will get this repetitive sequence that there will be one N, then C alpha, then C prime. This is the peptide bond. Then again, you see after this N, you get C alpha, C prime, right? And again, this peptide bond, this CONH, okay? Then again, you see this C alpha, the C prime, okay? So like that. Now, this peptide bond is a very interesting bond because what happens is it has a partial double bonded nature. This N has the lone pair electrons, so it forms this double bond and this opens here, right? It forms this negative charge. So it forms a partial, it has a partial double bond nature. So what are these dihedral angles? The dihedral angles are actually the angles of rotation of different planes, right? So these atoms, they are bonded um, because of the kind of sharing of these orbitals. The electronic densities, shared electronic densities are bonded, but they can rotate, right? The atoms can rotate around this bond. So basically, the planes can actually rotate. But this rotation of these different planes leads to different three-dimensional organizations or orientations of the amino acids, which in turn govern the structure that these amino acids take within the these protein secondary structural elements or tertiary structural elements. So that is what actually dihedral angles are. The angle of rotation between this N and C alpha is called the phi dihedral angle, and the angle of rotation between this C alpha and C prime is the psi rotational angle, dihedral rotational angle. But it's actually not this just this single amino acid residue. It's actually the four amino acid residues. For example, C alpha. If you just try to draw a plane, okay, this C alpha H and this C double bond O. So this plane, this is your one plane, okay, or basically actually this. Sorry, not this one. I'll just try to show you that this is one plane, okay. The C alpha, this plane and this plane. These are your two planes. Now this psi dihedral angle will be the angle of rotation between these two planes, right? Now, if you theoretically think of doing this, then you will say that any I can rotate it any angle is fine. I can rotate it to say any value of this phi, and all the values should be fine. But actually, that is not the case. The thing is, depending on what R group you have here, right? What R group you have here, depending on the bulkiness of the R group, there are some angles where these atoms come too close to one another, and there is something called a steric hindrance. For example, you are trying to fit 2%, you are trying to fit 5% in that seat of the bus, which is supposed to have 2%, right? There, what happens? There is a steric hindrance. You are facing such uncomfort, right? Facing such discomfort. So that is actually called steric hindrance. So that happens at certain angles. So those angles are not allowed. Those angles are kind of prohibited. Those conformations which lead to those angles are not energetically favorable. So those angles are not assumed. And this is why understanding this concept of dihedral angles is important, because there are, depending on this bulkiness of the R groups or the properties of the R groups, for example, it's positively charged, negatively charged, there can be positive repulsions and other things, steric hindrances, depending on which some angles, dihedral angles of arrangement of this of three-dimensional arrangement of these molecules is prohibited, whereas some are favored, which kind of forces them or drives them to assemble, to organize in certain orientations over others, thereby governing what fold the linear polypeptide chain takes eventually, depending on the amino acid composition, right? So why this amino acid composition is important is not only for the interactions that they can drive, like hydrogen bonding, hydrophobic interaction, van der Waals interaction, electrostatic interaction, but also for what phi and dihedral angles they can assume that govern their three-dimensional structure. Whereas it is very interesting to note and also very important to note that since I have already mentioned that this CN bond, right, which is basically the peptide bond, right, the C double bond and this C double bond O, the bond that connects the C double bond O and the NH. So this one, right, this is your omega bond, right? This is your omega bond. This one, this one. So this omega bond, which is actually the peptide bond, because of its partial double bonded nature, which is represented like this, right? Because of the partial double bonded nature, sharing of electrons. See, if you have a single bond, you can rotate easily, right? You can rotate very easily. But you have a double bond, right? You have to be, your rotation is immensely constrained. You can't rotate. If you rotate, then this double bond is not possible. So because of this partial double bond nature of this peptide bond, its rotation is highly restricted. It's almost a single plane, right? Which means that this angle, this omega angle, is strictly 180 degree. It's strictly constant, it's strictly planar, and it's 180 degree. Its omega doesn't change. So only what can change is phi and psi. These two bonds which join these two planes are allowed to rotate. But some angles again are not favorable because they cause steric hindrances or they cause positive repulsions or other repulsions or other energetically favorable conformations, energetically unfavorable or not favored conformations, right? Which are not assumed, whereas they can assume only certain phi psi values that are energetically favorable and that leads to their formation of a certain secondary structure and tertiary structure, thereby governing protein folding, right? That is what in a legendary piece of work, a scientist from India did, which was later recognized all over later recognized and appreciated, the due appreciation much later than his time. This was done by Ramachandran. So Ramachandran, much quite some several years ago, he asked he asked this question along with his students that can we model the protein structure from just the amino acid sequence? Now, for doing that, what we need to really understand is how a certain amino acid sequence leads to, or why a certain amino acid sequence leads to formation of a certain secondary structure, right? So that's where. So what he did is so this omega, which is the rotation of the omega is always 180, as I already mentioned, because it's the partial double bonded peptide bond, right? Partial double bond nature of the peptide bond, which prevents rotation. So it's almost like a single plane, so making the omega 180. So only phi and psi are allowed to be rotated. Only phi and psi can be rotated, can assume any value from 0 to 360. But what he found that that phi and psi cannot assume all the values. It assumes only a certain set of values. So because of which you can see that there are very high densities in these regions, right, which have a certain phi and psi values in these regions, right, in these regions. But there are a large number of phi and psi values, right? Each you have a certain phi value, certain psi value, right? But there are large spaces, large areas of phi psi coordinates which are unfavorable, right? Which cannot be assumed. These are energetically not favored for multitude of reasons. So this was kind of converting a 3D protein structure to a 2D map where you just took out the omega because the omega, due to the double bonded nature of the peptide bond, was leading to the formation of a single plane. And the three-dimensional structures were being governed by by these phi and psi values, by what phi and psi values can be assumed, can be um, formed by the amino acid residues. So later, so I mean, this is how it stands. Like this, this density was found that the amino acid residues that typically take this density of kind of these phi and psi values typically tend to form the alpha helices, whereas the amino acid residues which typically tend to assume those particular conformations and configurations that have these phi and psi angles typically form these beta sheets. So there is also protein breathing. There is a conformational flexibility and dynamics in protein also happening. So there is fluctuation within this set of phi and psi angles, but not to that phi and psi angle which lies in this region. These are unfavored. The empty spaces are unfavored, whereas you can see the densities lying in certain subsets, which actually stand for different secondary structural elements like beta sheets, alpha helices, polyproline 2 helices, turns, like right-handed alpha helices, left-handed alpha helices, 310 helices, etc. And some will also start. This polyproline helix will be your collagen helix, right? Um, yes, polyproline helix and collagen helix. Yeah. So all of these different densities having certain favorable sets of pi and psi angles actually lead to the formation of different secondary structural elements that Ramachandran mapped in this two-dimensional representation of three-dimensional protein fold, where one coordinate could be eliminated, which is omega, which was kept constant at 180 because of the planar nature of the peptide bond, the partial double bonded nature of the peptide bond. Okay, so almost this is what I had from my end. Um, Now we can be open to questions. If you have any, we can discuss them. No questions? Any question regarding the assignments that you need to do for this week? Okay, then. Thanks for joining. Then we will be meeting on the next Sunday or the tutorial of the third week. All the best for your assignments, and hope that you keep on learning new things. Hope you're enjoying the courses. Keep getting your doubts clarified as much as you can. We will try our best. Okay, all the best. Bye.